So there are three main stages of aerobic respiration, and stage one is glycolysis. Glyco meaning sugar and lysis meaning splitting. So glycolysis is the splitting of sugar. And what happens in glycolysis is glucose, which has got 6 carbons in it, is turned into pyruvate. 2 molecules of pyruvate, which has got 3 carbons in it. Now this takes 2 ATP at this end to produce 4 ATP at this end. And finally, it takes in 2 NAD plus to produce 2 reduced NAD, NADH. So, the point behind glycolysis is to split glucose. So the first thing is that glucose is isomerized into this isomer of glucose where you've got your 6 carbons all together. And then what happens is 2 ATP molecules, which are these things, these 2 ATP molecules are used to add phosphates to either end of the glucose molecule. And this is called phosphorylation. I'm not very good with beads. There we go.
You can tell.
That is phosphorylation. And what that leaves is hexose bisphosphate. You've got 6 carbons, hexose, but bisphosphate, 2 phosphates. Now this leaves us with 2 ADP, and our hexose bisphosphate is then split into 2 triose phosphates, because there's 3 carbons and 1 phosphate on those. Now the next stage is the arrival of 2 NAD with inorganic phosphates as these things, okay? And what then happens is that the TPs, 2 of them, so this happens twice, react with the NAD and the inorganic phosphates, and the phosphates bind to the TP to produce triose bisphosphate, because there are 2 phosphates but there's still only 3 carbons. And then this is slightly confusing with the beads, because obviously the beads are a simplification. The hydrogen molecules from inside these, from the triose phosphates, bind to the NAD to produce reduced NAD or NADH. And that bit is catalyzed by a dehydrogenase enzyme, and the NAD therefore serves as a coenzyme. Now, we can get rid of these because these aren't really relevant for a little while yet. And the final stage of glycolysis is turning these triose bisphosphate molecules into pyruvate by removing the 2 phosphates. And the way that works is 2 ADP for each of the triose bisphosphates. So that's basically the 2 that we already used plus 2 new ones. So we end up with a net increase of 2 ATP molecules. And the phosphates come off here through a series of reactions, which we won't need to go into, to produce 2 ATP, ATP, and a pyruvate from 1 triose bisphosphate, and on the other side we get another 2 ATP and another pyruvate. And so just to clarify at the end here, we'll bring back my awful little diagram, and we can see that we have got our 2 pyruvate produced from the glucose. We have turned 2 ATP into 4 ATP, which is pretty impressive, and we've turned 2 NAD into 2 NADH. So the second stage of respiration, that's respiration stage 2, is called the link reaction. The link reaction takes place in the matrix of the mitochondrion. So it takes place once the pyruvate, which we produced in glycolysis, gets across the membrane into the mitochondrion. And it's called the link reaction because it links glycolysis with the Krebs cycle. So here are our 2 pyruvate that we've got from glycolysis. And what happens in the link reaction is that there is an enzyme which is called decarboxylase, which separates off CO2 from each of the pyruvate. Now, you're just going to have to imagine the O2 molecules because I haven't got enough beads for them. But there you go, there's your CO2, and these are our 2 carbons left in our 2 carbon molecule. And at the same time that is happening, 2 NAD react with each of them, so 1 NAD with each 2 carbon molecule, and that takes away a hydrogen from each of these, which you've got to imagine was part of it to begin with. This is a simplification. And those bind together to form NADH, reduced NAD again. Stop rolling away, beads. And so we've got our 2 carbon dioxides and 2 reduced NADs. And what that leaves these 2 carbons as is acetate. Right, the final stage in the link reaction is the remaining 2 carbon molecules, that's your acetate, bind with an enzyme called CoA or coenzyme A. Okay? And that's these orange things, which I'm drawing in because I've run out of beads. And when the CoA binds with acetate, they become ACoA or acetyl coenzyme A, because you've got your acetyl group and you've got your coenzyme A. So the next stage in respiration, respiration stage 3, is called the Krebs cycle. And as the name implies, it is a cycle, so you can come in on this at any stage. Okay, we are going to come into the cycle where the acetyl-CoA, which is 2 carbons in the group here, binds with a 4 carbon molecule, which is called oxaloacetate, which is this molecule here, and these 2 bind together to form a 6 carbon molecule, which is called citrate. And that then leaves our coenzyme A group to go back off and pick up more pyruvate to convert into 2C to bring back into the Krebs cycle so the cycle can repeat. Okay, and now what happens is a series of redox reactions. So, first NAD comes in and takes off one of the hydrogen ion, a hydrogen which we've got to imagine as being a part of the citrate, and that then becomes reduced NAD or NADH. And so we've got 1 NAD goes off there, NADH goes off there, and 1 carbon comes off here into carbon dioxide. You've got to imagine the 2 oxygen there. And that leaves a 5 carbon molecule called alpha ketobutyrate. You don't need to know that. And each of these reactions is catalyzed by a different enzyme. Again, those you don't need to know the names of, you just need to know that they exist. And the next stage is basically the same thing happens again. So an NAD comes in and takes off one of the hydrogens, which we've got to imagine was there all along, produced reduced NAD, and a carbon comes off to form carbon dioxide, and that leaves us with a 4 carbon compound which is called succinate. At this stage ADP comes in and is phosphorylated, and this is substrate level phosphorylation because there's no movement of protons. It is phosphorylated into ATP, and this is the only time that ATP is actually directly produced in the Krebs cycle, which might make it seem like it's not very efficient going through all this process just to produce one ATP molecule. But actually what it's producing, the reduced NAD and in a minute the reduced FAD, is then used to produce much more ATP in a later stage. And the ATP can float off and do its thing. Okay, so now that the ATP has diffused away, what we're left with is another 4 carbon molecule which is called fumarate. And what happens next is FAD comes in here, and what happens to the FAD is basically the exact same thing that happens to the NAD. It is reduced by a hydrogen atom coming off the 4 carbon molecule and binding with it to produce reduced FAD or FADH, and that moves away. And then we have the exact same thing, a reduction reaction taking place on the on another NAD molecule, which comes in here. And so our maltate, as it now is, then loses another hydrogen to go with this NAD to produce reduced NAD. And what that leaves us with is oxaloacetate, which, if you remember, is what we started the Krebs cycle with all that time ago. Okay, so the final stage of respiration takes place on the cristae, which is the membrane in between the intermembrane space and the matrix. It's the inner membrane of the mitochondria. And this is where most of the ATP is produced in respiration, and it's where we're going to use our NADH and our FADH, which we produced earlier. And basically what it is it's just a series of redox reactions. So embedded in the membrane are these protein carriers, and they're going to be very important. So if we take our NADH and it comes up to the first protein carrier, and what happens here is it has the hydrogen removed from it, and that releases NAD, and the NAD plus, I should say, and the hydrogen splits in 2, and that produces a H plus ion, or a proton, and it produces an electron. Now the proton, because obviously there's only one of them in a hydrogen, goes through the carrier protein into the intermembrane space. And this happens many times because we're going to have lots and lots of NAD coming up here, popping open, and you have your NAD floating off here, and our hydrogen is split, and the protons go up here. And the electrons are transported along the electron carrier chain through all of these to the final electron receptor, which is that oxygen molecule there. Okay? And at the same time, the same thing is happening to reduced FAD. So the H is popping off, it's splitting in 2, and the electrons are going up here, around here, and into our oxygen molecule. And what we've also got then now is we're going to imagine that these blue things are H plus, they're protons for the time being. They're building up here, and we've got all our electrons building up here. And what that means is we've got this massive electrochemical gradient. And we've got no way for the hydrogen ions, for the protons, to get back across, because obviously they're polar, so they can't get through this membrane, and they can't go back through these protein carriers. The only thing they can get through is this ATP synthase, which is a very cool molecule which spins in a particular way to help produce ATP. So using the energy produced by the protons coming through here, setting the whole thing spinning very awesomely, we can phosphorylate these ATP molecules into ATP. Here we go. And this is happening many, many times a second with all of the different protons coming from the NAD plus, from the reduced NAD I should say, and from the reduced FAD. And the final bit, which makes the whole thing work, is these H plus ions, the protons, have come through here. We've got all our electrons in here, which you're just going to have to imagine because I haven't got beads for them. And so the H plus and the electrons and the oxygen, which has accepted the electrons from here, look, because it's the final electron acceptor, bind together, and that gives us H2O. And what is H2O, William?
Water?
Thank goodness you got that right. That would have been embarrassing if you got it wrong, wouldn't it?
I thought it might be a trick question.
So, William, do you now understand vaguely how respiration works?
Not a clue.
No, me neither.